Medical parasitology. Parasitology deals with a parasite which infects and produces disease in human beings. A parasite is a living organism which depends on the living host for its survival and derive nutrients from the host without giving any benefits to the host. It is defined as an organism which harbors the parasite. Different types of host or as follows: a definitive host. It is the host that harbors the adult stage of a parasite or where the parasite replicates sexually. Intermediate host. It is the host that harbors the larval stage of the parasite or where the parasite replicates asexually. Reservoir host. It is the host that harbors the parasite and acts as an important source of infections. Host parasite relationship, an association which is formed between the host and the parasite, may be divided into following types. Symbiosis. Both host and parasite are so dependent upon each other that one cannot live without the help of the other. None of them suffers any harm from the associations. Commensalism. It is the association in which the parasite only is deriving benefit without causing any injury to the host. A commensal is capable of living an independent life. Parasitism. It is an association in which the parasite derived benefits and the host gets nothing in return and always suffers some injury. The parasite cannot live in an independent life. Classification of parasites. Parasites are classified into protozoa, helminthiae. A classification of medical importance is given in the table. Protozoa: unicellular, amoeba, Entamoeba histolytica, Entamoeba gingivalis, flagellates, Giardia, Lamblia, Trichomonas vaginalis, Leishmania species, Sporozoa, Plasmodium vivax, Plasmodium falciparum, Toxoplasma gondii, Ciliates, Balantidium coli, Helminthes, multicellular, Trypanotodes, Fascicola species, Cystosoma species, Cystodes, Taenia species, Echinococcus, nematodes, roundworms, hookworms, and threadworms. Protozoa: Entamoeba histolytica. Entamoeba histolytica is worldwide in distribution but more common in tropical and subtropical countries. It lives in large intestine of man. Morphology: the parasite exists in three forms: tropozoid, pre-cystic stage, and cyst. Tropozoids. It measures 18 to 40 micrometer, average 20 to 30 micrometer diameter. The cytoplasm of the tropozoids is divided into two portions: a clear outer ectoplasm and a granular endoplasm. The tropozoids are mottled by pseudopodia. Pseudopodia are long finger-like projections of ectoplasm into which the endoplasm flows. The nucleus of a tropozoid is spherical in shape and varies in size from 4 to 6 micrometer. In stained preparation, it shows a small dot-like structure, karyosomes, which is central in position. Pre-cystic stage. It is smaller in size and varies from 10 to 20 micrometer diameter. It is round or oval with a blunt pseudopodia. Nucleus has a similar characteristics as that of tropozoid. Cyst. It is around 10 to 15 micrometer in diameter and it is surrounded by a highly refractile membrane called cyst wall. The nuclear structure is similar to that of a tropozoid. A mature cyst is a quadrinucleated spherical body but soon divides by the binary fission to form 2 and then 4 nuclei. Uninucleated and binucleated bodies also possesses glycogen mosses and 1 to 4 chromatial or chromatoid bars. The glycogen moss stained brown with iodine. Chromatoid bars do not stain with iodine but appears as a refractile bars in preparation with normal saline. In a mature cyst quadrinucleate, both the glycogen moss and chromatoid bars disappears. Life cycle. Entamoeba histolytica passes its life cycle in only one host. The mature quadrinucleated cyst or the infected form. Man acquires the infection by ingestion of water and food containing the cyst. When the cyst reaches the cecum or the lower part of the ileum, the excistation occurs. During this process, each mature cyst liberates a single amoeba with 4 nuclei. The tetranucleated amoeba which eventually produces 8 metacyst tropozoids by the division of nuclei by binary fission. These metacyst tropozoids ultimately lodge in the submucosa tissue of the large intestine, their normal habitat. Here they grow and multiply by binary fission. During growth, Entamoeba histolytica secretes a proteolytic enzymes which brings about destruction and necrosis of tissue, leading to flask-shaped ulcer. A large number of tropozoids are excreted along with the blood and mucus in the feces. This condition is called amoebic dysentery. Sometimes the tropozoid enters into the radicals of portal veins to be carried away by the liver. In the liver, they multiply and produces amoebic hepatitis and amoebic liver abscesses. After sometimes when the effect of the parasite on the host is toned down and there is increase in the tolerance of the host, the lesion starts healing. The tropozoites in the lumen of a large intestine transforms into the pre-cyst and then into mature quadrinucleated cyst. This process is called the encystation. Diseases: amoebic dysentery, intestinal amoebiosis, incubation period 1 to 4 weeks, ulcerative lesions or profuse bloody diarrhea, extra-intestinal amoebiosis, the hepatic amoebiosis or amoebic liver abscesses. The amoebic liver abscess may occur in any part of the liver, but it is generally confined to posterior superior surface of the right lobe of the liver. The pus of the liver abscesses has a characteristic red-brown anchory sauce appearance. Other organs such as lungs, brain, spleen, skin may also be involved. Laboratory diagnosis. The laboratory diagnosis depends on the demonstration of entamoeba histolytica in material obtained from any particular lesions such as stool, pus from the hepatic abscesses cases and spotums. Intestinal amoebiosis stool examination. In acute amoebic dysentery, stool or colic scrapings from the ulcer are examined by naked eye and microscopy examination. The normal saline preparation is useful for the demonstration of actively motile tropozoites, while the iron preparation is required for the study of cyst or dead tropozoites. The excretion of cyst in the stool is often intermittent, therefore at least 3 consecutive specimens should be examined. Charcoal laden crystals may appear in the saline preparations. These are the diamond-shaped crystals, clear and refracted. Amoebic dysentery must be differentiated from bacillary dysentery. B. In blood exam, it shows leukocytosis. Serological test. In early cases, it is always negative. The antibody can be detected in the later stages of invasive intestinal mucosis. The serological diagnosis used or indirect hemagglutination assay and enzyme link immunosorbent assay. Hepatic amoebiosis. Diagnostic aspiration. Tropozoites of entamoeba histolytica may be demonstrated by microscopy of pus aspirate from the hepatic abscesses. Liver biopsy. The tropozoites may also be demonstrated in the liver biopsy from cases of amoebic hepatitis. Examination of blood. It shows leukocytosis. Stool examination. In about 15% of hepatic cases, the cyst of entamoeba histolytica may be demonstrated in the stool. It indicates persistent intestinal infections. Serological test. The antibody detection is of great value in the diagnosis of hepatic amoebiosis. IHA and ELISA. ELISA are some serological tests used for serological diagnosis. A serum with antibody titer of 1:128 or more by IHA is diagnostic for amoebic hepatitis. Entamoeba histolytica region can also be detected in the serum by ELISA. Entamoeba coli. It is a worldwide in distribution. It lives in the lumen of large intestine. It is not pathogenic. Life cycle of E. coli is similar to that of Entamoeba histolytica. The Entamoeba coli also exists in 3 stages: tropozoid, pre-cystic stage, and the cyst. Difference between the Entamoeba histolytica and Entamoeba coli also shown in the table. Giardia lambelia. These flagellates inhabit the intestine and are therefore named as intestinal flagellates. They are present in duodenum and upper part of the jejunum of man. Morphology: Giardia lambelia exists in 2 phases: tropozoid and cyst. Figure 67.3. The shape of the tropozoid is like that of a tennis or a badminton racket. The dorsal surface is convex while the ventral surface is concave, having a suckling disc. Morphology: Giardia lambelia exists in 2 phases: tropozoid and cyst. The shape of the tropozoid is like that of a tennis or badminton racket. The dorsal surface is convex while the ventral surface is concave, having a suckling disc. The size of the tropozoid is 14 micrometer long and 7 micrometer broad. The anterior end is rounded and broad while the posterior end is pointed. It is bilateral, symmetrical. That is, all the organs of the body are paired and hence they are 2 exostyles, 2 nuclei and 4 pairs of flagella. The cyst is oval in shape and it is 12 micrometer long and 7 micrometer broad. There are 4 nuclei. The axostyle lie diagonally. The remnant of the flagella and the suckling disc may be seen in the cytoplasm life cycle. The tropozoid of the parasite multiplies in the intestine of the man by binary fission. Life cycle is depicted in the form of diagram. Disease: the mal absorption of fat, looseness of bowel, and mild stenuria. The patient may present as a silent case without any symptoms, as chronic enteritis and acute enterocolitis or chronic cholecystitis. Laboratory diagnosis: microscope examination. The demonstration of giardia, trophocytes, and cysts is freshly passed stool by a wet preparation, saline or iodine. Trophozoid may also be recovered in bile. Leishmania donovani. This parasite causes a visceral leishmaniasis or cholera. It is a parasite of a reticuloendothelial system. It is endemic in many places of India. Morphology: the parasite exists in two forms. Amastigote occurs in man. Promastigote occurs in gut of sandfly and artificial culture. Amastigote stages. It is a flagella stage, and it is present in the cells of a reticuloendothelial system of a vertebrate host, man, dog. It is a round and oval bodies. Promastigote form. It is a flagellar stage, and it is present in the insect vector and in culture. It is a long, slender, spindle-shaped body. 15 to 20 micrometer into 1 to 2 micrometer. Life cycle: the amastigote form multiplies in the cells of reticuloendothelial system till the host cell becomes packed with the parasite. The host cells enlarges and eventually ruptures. The blood-sucking insect sandfly draws these three free amastigote form during its blood meal. These amastigote forms develop into the promastigote form which again multiplies by the binary fission, producing a large number of promastigotes. The sandfly species named Plebotomus argentipus acts as an Indian vector. The infection occurs by the bite of infected sandfly. The incubation period varies from 3 to 6 months. Disease: the causes are visceral leishmaniasis. It is a characteristic of pyrexia, splenomegaly, hepatomegaly, lymphadenopathy, or general features of anemia. Laboratory diagnosis of causes depends upon the direct evidence and indirect evidence. Direct evidence demonstrates leishmania donovani in specimen. Direct evidence: peripheral blood smear. Thick blood film is examined for demonstration of amastigote form. Blood culture can be done in NNN Novi McNeil Nicody medium and incubated at 22 to 24 degrees centigrade. Promastigote form can be demonstrated. Biopsy: amastigote form can be demonstrated in the stained film. The promastigote form can be demonstrated when this material is cultured in NNN medium. The splenic puncture: it is one of the most valuable methods for diagnosis. Amastigote are found in stained films and promastigote in culture. The only risk of a splenic puncture is that bleeding may continue for the puncture wound results in the death. Indirect evidence: blood count. The progressive leukopenia: the proportions of leukocyte to erythrocyte is greatly altered. Hemoglobin estimation: anemia. Non-specific serological test: RDA test. It shows increased serum gamma globins, and thus in non-specific antimony test it is also dependent upon the increased serum gamma globulin. Complement fixation test with a WKK antigen. It detects antibody in the serum of a Kala Azar patient. The antigen used or prepared for human tubercle bacilli. The test is not specific since the antigen is not prepared from Leishmania donovani. Specific test: indirect hemoglobin assay, indirect fluorescent antibody test, and enzyme link immunosorbent assay or more specific test. The ELISA uses using species-specific monoclonal antibody and DNA probe are very useful in the direct detection of Leishmania antigens. Plasmodium: there are 4 species of genus Plasmodium: Plasmodium vivax, Plasmodium falciparum, Plasmodium malariae, and Plasmodium ovale. They are protozoa causing malaria in man. In India, Plasmodium vivax, Plasmodium falciparum are very common, but Plasmodium ovale does not occur. Life cycle: the malaria parasite passes the life cycles in 2 phases, that is man, intermediate host, and female, Anopheles mosquito, definite host. Asexual development of a parasite occurs in man and sexual development in mosquito. Human cycle: the spermatozoid spermatozoid is the infected form of the malarial parasite. These spermatozoids are present in the salivary gland of female Anopheles mosquito. Man gets infected by the bite of infected mosquito, and sporozoid are introduced directly into the blood circulation. Thus the human cycle starts, and it comprises of the following stages: pro-erythrocytic Schizogony, erythrocytic schizogony, gametogony, exo-erythrocytic schizogony, pre-erythrocytic schizogony. Before starting erythrocytic schizogony in the blood, the sporocyte undergoes a development phase inside the liver cells. This phase is a development, and it is known as the pre-erythrocytic schizogony. The sporocyte, elongated and spindle shape, becomes rounded inside the liver parenchyma cells. They undergo multiple nuclear divisions and develop into the schizoid. The size of the schizoid varies in different species, and it contains 20,000 to 50,000 merozoites. The pre-erythrocytic cycle lasts for 8 days in Plasmodium vivax, 6 days in Plasmodium falciparum, 13 to 16 days in Plasmodium malaria, 9 days in Plasmodium ovale. After completion of the cycle, the liver cells ruptures and releases merozoite into the bloodstream. Erythrocytic schizogony. The merozoitic release from the pre-erythrocytic schizogony penetrates red blood cells. They passes through the stage of tropozoites, schizoids, and merozoites. Depending on the species of malarial pulses, they may be 6 to 24 merozoite in the red blood cells. The red blood cells ruptures to release the merozoite, which attacks the new red cells and continues the erythrocytic schizogony. Repeated the cycle. In Plasmodium falciparum infection, erythrocytic schizogones aggregates in the capillary of the brain and other internal organs. So the only ring formed are found in the peripheral blood. Differential features of various Plasmodium are shown in the table. Gametogony. Some merozoid or erythrocytic schizogony developed into a male and female gametocyte known as microgametocytes and macrogametocytes respectively. They developed in the red blood cells. These are sexual forms and are found in peripheral blood. The microgametocyte of all the four species are small in size. Cytoplasm stained light blue, and nucleus is large and diffuse. In contrast to macrogametocyte or large, the cytoplasm stained deep blue and the nucleus is small and compact. Differential features of Plasmodium in man feature form in peripheral blood. Plasmodium vivax, tropozoid, schizont, and gametocyte. Plasmodium falciparum, ring form and gametocyte, crescent shape. Plasmodium malaria, tropozoids, schizont, and gametocyte. Plasmodium ovale, tropozoid, schizont, and gametocyte. 2. Yearly tropozoid or ring form. Plasmodium vivax 2 to 5 micrometer in diameter. Cytoplasm opposite the nuclear is thicker. Plasmodium falciparum 1 to 1.25 to 1.5 micrometer in diameter. Multiple ring in one ring blood cells forms ocule. Plasmodium malaria similar to that of Plasmodium vivax. Plasmodium ovale similar to that of Plasmodium vivax. Tropozoid 3. Tropozoid Plasmodium vivax irregular amoeboid vacuoles present. Plasmodium falciparum compact form, rarely amoeboid pigment collect into the single mosses. Plasmodium malaria band shape, single amoeboid vacuole inconspicuous. Plasmodium ovale compact, not amoeboid vacuoles inconspicuous. Schizoid Plasmodium vivax 9 to 10 micrometer in diameter, almost completely fills and enlarges erythrocyte. Plasmodium falciparum 4.5 to 5 micrometer in diameter, fills 2/3 of normal size erythrocyte. Plasmodium malaria for 6.5 to 7 micrometer in diameter, almost fills a normal size erythrocyte. Plasmodium ovale 6.2 micrometer in diameter, fills about 3/4 of slightly enlarged erythrocyte. Merozoids Plasmodium vivax 12 to 24 in diameter. Plasmodium falciparum 18 to 24 in number. Plasmodium malaria 6 to 12 in number, and Plasmodium ovale 6 to 12 in number. Gametocytes in Plasmodium vivax spherical, much larger than red blood cells. Plasmodium falciparum crescentric sickle shape larger than a red blood cells, Plasmodium malaria, round size of red blood cells, Plasmodium ovale, round size of the red blood cells, female gametocyte, macro gametocyte, spherical, large than male gametocyte, cytoplasm strains, deep blue, nucleus is small and compact, Plasmodium falciparum, sickle cell longer, more slender, cytoplasm stains deep blue, nucleus in is compact, Plasmodium malaria similar to the Plasmodium vivax but smaller, Plasmodium ovale similar to that of Plasmodium vivax but smaller, male gametocyte micro gametocytic, Plasmodium vivax is spherical, smaller than female gametocyte, the cytoplasm stain light blue or pale blue, nucleus is large and diffuse, Plasmodium falciparum sickle shape, broader, shorter, cytoplasm stains light blue, nucleus is diffuse, Plasmodium malaria similar to that Plasmodium vivax but smaller, Plasmodium ovale similar to that of Plasmodium vivax but smaller, malarial pigment, Plasmodium vivax yellowish brown, Plasmodium falciparum dark brown, Plasmodium malaria dark brown, Plasmodium ovale dark yellowish brown, infected erythrocyte, Plasmodium vivax enlarged pale Schuffner's dots present, Plasmodium falciparum size unaltered, Maurer's dot present, Plasmodium malariae normal size and no dots, Zeeman's dot or prolonged staining, Plasmodium ovale slightly enlarged oval shaped and James dots present, ages of erythrocyte infected, Plasmodium vivax young, Plasmodium falciparum all ages young and old, Plasmodium malaria old, Plasmodium ovale young, the duration of erythrocytic schizoid, Plasmodium vivax 48 hours, Plasmodium falciparum 48 hours, Plasmodium malaria 72 hours, and Plasmodium ovale 48 hours. Erythrocytic schizogony. The erythrocytic cycle resembles the pre-erythrocytic cycle. Some sporozoite on entering into the liver cells do not undergo asexual multiplication but enters into a resting dormant phase. The resting phase of parasite is known as a hypnozoids. After some period, usually up to 2 years, the hypnozoids reactivate to become the schizoids and releases a merozoids. These merozoites attack the red blood cells and are responsible for lapse of malaria. The exoerythrocytic schizoid is present in the Plasmodium falciparum. Therefore, lapse do not occur in malaria caused by the Plasmodium falciparum. Mosquito cycle. The sexual cycle of malarial parasite actually starts in the human host by formation of gametocyte, which are then transferred to mosquito for further development. A female anopheles during its blood meal from the patient ingested both the asexual and sexual form of the parasite. Only the mature sexual form are capable of further development in the mosquito and rest die immediately. In the midgut of mosquito, one microgametocyte develops into 4 to 8 thread-like filamentous structure named the microgametes. From one macrogametocyte, only one macrogamete is formed. The fertilization occurs when the microgamete penetrates into the macrogametes. The fertilized macrogamete is known as a zygote. The zygote lengthens and matures into the ookinete. The ookinete developed into the oocyte. As oocyte matures, it may increases in size and large number of sporocytes, a few hundreds to thousand, developed inside it. The number of oocytes in the stomach wall varies from a few to more than hundreds. The oocyte ruptures and releases a sporocyte in the blood in the body cavity of a mosquito. The sporozoites are distributed into the various organs and tissue of the mosquito. However, they have a special predilection for salivary glands. Mosquito is now capable of transmitting the infection to a man. Disease. The infection with the Plasmodium causes intermittent fever, which is named as malaria. The incubation period is 10 to 14 days in Plasmodium vivax, Plasmodium falciparum, Plasmodium ovale, but is 20 to 30 days in Plasmodium malaria. The typical clinical features consist consists of a febrile paroxysm followed by the anemia and splenomegaly. The Plasmodium falciparum is the most pathogenic of Plasmodium species of infecting man. There are no relapse in Plasmodium falciparum infection, but relapse occurs in Plasmodium vivax infection. The Plasmodium falciparum invades erythrocyte to all ages, young and old, but Plasmodium vivax infect only younger erythrocyte. The complication of a Plasmodium falciparum infection includes pernicious malaria and black water fever. The pernicious malaria is life-threatening complication that sometimes occurs in acute falciparum malaria. Various manifestations of pernicious malaria are grouped as cerebral malaria, alleged malaria, and septicemic malaria. The black water fever is a manifestation of infections with the Plasmodium falciparum occurring in those people who have been previously infected and have had an inadequate dose of quinine. It is characterized by intravascular hemolysis, fever, and hemoglobinuria. Laboratory diagnosis: light microscope. The microscopic examination of peripheral blood smear is the most important diagnostic procedure in the malaria. Thick and thin smear of blood are prepared on the same slide or two different slides. Blood for smear should be collected a few hours after the height of febrile paroxysm because the parasites are most abundant during this period. The blood is taken by the pricking a finger prior to start the anti-malarial therapy. For thick smear, take a drop of blood on the slide and spread it in an area of 1 square centimeter. For thin smear, take a drop of blood at one corner of the slide and spread it with another slide so that the tail formation occurs at another end. Both the thick and thin peripheral blood smears are stained with Leishman stain. These smears are examined under oil immersion lens. Malarial parasite may be quickly detected in the thick smear, but species identification is difficult. The thin smear is examined for identification of the species of Plasmodium. The parasites are more along the upper and lower margin of the tail of the thin smear. A minimum of 100 microscopic field should be examined in this slide. All asexual erythrocytic stages, ring form tropozoites, schizoids, as well as gametocytes can be found in the peripheral blood smear in infection with Plasmodium vivax, Plasmodium malaria, Plasmodium ovale. But in Plasmodium falciparum infections, only the ring forms and gametocyte crescent shape can be seen. The multiple ring in an individual red cell with acole forms in its diagnosis of Plasmodium falciparum. The red blood cells are enlarged in Plasmodium vivax, but there is no enlargement in RBC in Plasmodium falciparum infections. Fluorescent microscope. The blood smear is made on a slide and it's stained with acridine orange. The stained slide are examined with a fluorescent microscope. Nuclear DNA of malarial parasite is stained green and cytoplasmic RNA red. Quantitative Buffy coat QBC test. The blood is collected in a QBC tube coated with a fluorescent dye and it is subjected to centrifugation. After centrifugation, the Buffy coat in the tube is examined directly under the fluorescent microscope. The acridine origin stained malarial parasite appears brilliant green. Rapid diagnostics: detection of antigen using immunochromatographic method. The test is available to detect the histidine-rich protein HRP2 of Plasmodium falciparum and parasite lactate dehydrogenase of all 4 Plasmodium species that infects human. Serological test: indirect immunofluorescence test, indirect heme agglutination assay, enzyme link immunosorbent assay, DNA probe. Treatment: the chloroquine is used for treatment of acute malaria. Mefloquine is active against chloroquine resistant strain. Trematoids. These are leaf-shaped unsegmented worm called flukes. Each individual worm is a hermaphrodites except the schizosomes, which are unisexual. The body cavity is absent. The worm is oviparous. Eggs are liberated. Eggs are all operculated except those of schistosomes and developed into water. Man harbor adult worm, eggs liberated and developed in the water, passes through different stages in the snail. Cessation free in water, cessation on circarian on vegetables or within second intermediate host. The circarian free in water enters in the human and developed into adult worm. The circarian or on vegetables or within second intermediate host ingest and develop into adult worm. Different trematoids, intestinal trematoid example fascicola busci, hepatic trematoids example fascicola hepatica, lung trematoids example paragonium westermanni, blood trematoids example cystosoma hematobium, cystosoma mansoni, cystosoma japonicum. Mode of infection by ingestion of encysted cercariae in vegetables. The F. hepatica and F. busci, the fascicola hepatica, fascicola busci, fish or flesh of crab, the Pergonimus westermanni, free circarian penetration through the skin. Example, cystosoma hematobium, cystosoma mansoni. Cystodes, Tinea saginata, the common name of Tinea saginata or beef tapeworm, and the unarmed tapeworm. It is worldwide in distribution. The infection occurs by eating raw or insufficient cooked beef. In India, they are particularly prevalent among Mohammedans, but it is not found in Hindus who do not eat beef. Morphology, adult worm, it measures 5 to 10 meter in length, but may be up to 24 meters. It is white or semi-transparent. It consists of three parts: scolex, neck, and proglottis. The scolex or head is quadrated in shape and has four circular suckers which may be pigmented. It measures 1 to 2 millimeter in diameter. The head does not contain any rostridium or hooklets. The neck is long and narrow, about 0.5 millimeter in width. The proglottis or segment are 1000 to 2000 in number. The length of the gravid segment is 3 to 4 times in breadth. Eggs. The eggs are liberated by the rupture of rib proglottis. The characteristic feature of egg, spherical or bile stained, 31 to 43 micrometer in diameter. The inner embryo force is thick-walled, radially striated and brown in color. It contains oncosphere measuring 14 to 20 micrometer diameter, containing 3 pairs of hooklets. It does not float in the saturated solution of common salt. It is infective only in cattle. Tinea sodium, common name of Tinea sodium or pork tapeworm and armed tapeworm. It is also worldwide in this distribution. The infection occurs by eating raw or insufficient cooked pork. It is uncommon to Mohammedans who are not generally pork eaters. Morphology, adult worms, it measures 2 to 3 meter in length. It also consists of 3 parts: scolex, neck, and proglottis. The scolex or neck is a globular in shape and has 4 circular suckers. The head contains a rostidium armed with a double row of hooklets. The neck is short and measured 5 to 10 millimeter in length. The proglottis or segment are 800 to 900, less than 1000 in numbers. Eggs, the characteristic feature are the same as those of Listeria Tinea saginata. The eggs are infective to both pig and man. Life cycle. The worm passes its life cycle in two hosts, the definitive host and the intermediate host. The man is a definitive host which harbors the adult worm, and the pig is intermediate host which harbors the larval stage. The adult worm lives in a small intestine of man, and the eggs or gravid proglottis are passed out with the feces. The animal becomes infected by swallowing these eggs. After ingestion, oncosphere hatches out and penetrates in the gut wall. It is carried by in a systemic circulation. The naked oncosphere are filtered out from the circulation of blood into the muscular tissue. They are transformed into the cysticerous stage in the muscles when these are known as a circus cellulosa. The human being the infected by eating uncooked or partially cooked pork containing circus cellulosa inside the alimentary canal of man. The scolex of the circus avaginates and attaches to the gut wall by suckers and then developed into the adult worm. Life cycle of Tinea saginata is the same except that intermediate host is cattle and larval stage is named as a Cysticercus bovis.